Dear student, in this video, we study self-incompatibility in plants. This is included in your paper number 9, Genetics and Plant Breeding, and point number 3.1. In this video, we study the types of self-incompatibility, the use of or importance of these self-incompatibility in plants. There are two types of self-incompatibility. First is gametophytic and second one is the sporophytic self-incompatibility. In case of a gametophytic, which is labeled as a GSI system, in this sigma is smooth and weight. In this sporophytic, which is labeled as a SSI system, in this stigma is a papillate, means it is a hairy one and it is a totally dry one. In case of a GSI system, outcome of the interaction between the pollen tube and the style is determined by the genotype of the pollen gamete. Means in this gametophytic system, the genotype of that is determined by only the genotype of a pollen gamete. It is a very important one, therefore it is termed as a gametophytic system. Gamete means pollen grain is responsible for the determination of that genotype. While in case of a sporophytic system, outcome of that interaction between the pollen tube and the style is determined by the genotype and sporophyte. Genotype of the sporophyte means that sporophytic tissue is a diploid tissue and it determines the genotype, and therefore this interaction which is takes place in between pollen tube and style, that interaction is only determined by the genotype of a sporophyte, therefore it is termed as a sporophytic self-incompatibility or simply SSI system. In case of a gametophytic system, the S locus products are synthesized after completion of meiosis, while in SSI system, S locus products are synthesized before completion of meiosis. Because this S locus is nothing but a site where the gene which is located, and that gene is nothing but this S1. S gene is present, and that S gene is mainly responsible for the formation of these multiple alleles S1, S2, S3. These are nothing but S3, S4. These are nothing but multiple alleles which is produced from that S locus, and that S locus products means production of these S1, S2, S3, S4. These local products, locus products, these are synthesized after completion of meiosis. When meiosis is completed, at that time these S locus products, these are synthesized. While in case of a SSI system, the S locus products are termed as synthesized before completion of meiosis, because these are mainly determined by the genotype of a sporophytic tissue. Therefore, in them there is no need of a meiosis. Before that, S locus product means S, the gene, these are synthesized, that S1, S2, S3, S4. These are number of alleles from that S locus before the meiosis is that takes place. Before the completion of a meiosis, that production of these S locus, this we observe in case of a sporophytic tissue self-incompatibility system. Then here, in case of a gametophytic system, gametophytic incompatibility, growth of the pollen tube arrest in the style. The growth of pollen tube which is takes place, it is arrested in that in the style. This is nothing but a style. These are nothing but a stigmatic lobe, and this black color shown here, these are nothing but a pollen grains. Means in this gametophytic system, last lecture I told you that the cell incompatibility is nothing but a thing due to which the seed setting does not take place in that plant, and there are several reasons. Due to that reasons, these seed setting in that plant is not possible, and in that first one is nothing but the stigmatic lobe. Here it is a dry one, and due to which the pollen grains do not germinate. If a pollen grains is a germinate, then their style, or sorry, their pollen tube does not immediately towards the ovule which is present in the ovary. If the growth of that pollen tube is a slow one, then the maturation period of ovule is a pass on, and therefore in that case there is not a proper fertilization, seed setting is takes place, and therefore in that gametophytic system we found that here growth of pollen tube, these are arrested in the style. Growth of pollen tube is arrested in the style, therefore it is termed as a gametophytic system. While in case of a sporophytic system, growth of the pollen tube arrests at the surface of a stigma. Only at the surface of a stigma, the growth of that pollen tube is arrested. Then there are again a two system, homomorphic system as well as a heteromorphic system. His homomorphic system, in this homomorphic system, incompatibility is not associated with morphological differences among flowers. The incompatibility reaction of pollen may be controlled by first the genotype of the sporophyte, that is the plant on which it is produced, and it is coming under the sporophytic control. And the second, the own genotype of male gamete, that is nothing but of a pollen grain, and therefore it is termed as a gametophytic control. In homomorphic flowers, all flowers have exactly the same structure. Avoidance of self-fertilization depends on the genetic or biochemical mechanism, and on that basis there are these two type of system, that is sporophytic self-incompatibility, and second one is nothing but a gametophytic self-incompatibility. Means in these sporophytic self-incompatibility as well as a gametophytic self-incompatibility, as we study that this incompatibility reaction in case of a SSI system, it is takes place in between pollen tube and the style, and that is determined by the genotype of the sporophytic, that is the diploid tissue. Then growth of the pollen tube is arrested at the surface of the stigma, and S locus products are synthesized after completion of meiosis. This thing is takes place in a sporophytic self-incompatibility, while in case of a gametophytic self-incompatibility, the S locus products are synthesized before completion of a meiosis, and this incompatibility reaction between the pollen tube and the style is determined by the genotype of the pollen, and pollen is nothing but a haploid tissue, therefore it is termed as a gametophytic. Sporophyte is a diploid one, correct? You remember these two point. And growth of the pollen tube in case of a gametophytic self-incompatibility is arrested in the style. Then sporophytic self-incompatibility, the stigma is a papillate, that is nothing but a hairy one, and it is a dry one, and it is covered by hydrated layers of proteins, and therefore this protein layer is known as a pellicle. Within few minutes of reaching the stigmatic surface, the pollen releases an exine exudate, which is either proteinous one as well as it is made up of a glycoprotein. This exudate includes immediately the callose formation in the papillae of incompatible stigma, and thus in sporophytic system, stigma in the site of incompatibility reaction. Once the pollen tube crosses the stigmatic barrier, there is not further inhibition of the pollen tube growth. And this form of self-incompatibility has been studied intensively in the members of a mustard family. Means in the family Brassicaceae, this sporophytic self-incompatibility is studied, and again other example is turnips, rapeseed, cabbage, broccoli as well as in a cauliflower. While biochemical mechanism of gametophytic self-incompatibility, that is the GSI system, in that case the inhibiting the germination of a pollen on the stigma and inhibiting the elongation of a pollen tube in the style. Means in that system the germination of a pollen tubes on that stigma does not take place. Or second point, if that pollen grain is germinating, then the pollen tube, their elongation does not occur through, which is passes through that style. And these mechanisms are based on protein-protein interaction, and the best understood mechanism are controlled by the single S gene. And that S gene is termed as a S locus, which has a mainly different allele in the species population. Despite their similar morphological and genetical manifestations, these mechanisms have evolved independently and are based on different cellular components. Therefore, each mechanism has its own unique S gene system. And on that basis, these S locus contain the two basic protein coating region, which is labeled as expressed in the pistil and second expressed in the anther or in a pollen. Means first expressed in pistil it is a female determinant, and expressed in anther it is a male determinant. Because of their physical closeness, these S loci are genetically linked and are inherited together as a single unit. These units are called as S haplotypes. As a result of interaction of the protein encoded by the two regions of the S locus of the same haplotype, the pollen germination or pollen tube elongation is arrested, and it is responsible for the preventing the fertilization. However, when a female determinant interacts with a male determinant of a different haplotype, no self-incompatibility occurs and fertilization ensues. This is a simplistic description of the general mechanism of self-incompatibility. Following is the detailed description of the different known mechanism of a self-incompatibility in plants, in that in a gametophytic self-incompatibility, that is a GSI, the self-incompatibility phenotype of the pollen is determined by its own gametophytic haploid genotype. This is a more common in the type of self-incompatibility existing in the families such as the Solanaceae, Rosaceae as well as the Papaveraceae. Then there is a significance of this self-incompatibility in that first, nature has got balanced inbreeding and outbreeding regulated by interspecific and intraspecific incompatibilities. Interspecific means two different species in them that incompatibility is present, and intraspecific means in a single species this incompatibility is present. Therefore, the significance of that first incompatibility, self-incompatibility is it is having the balanced inbreeding and outbreeding, which is regulated by interspecific and intraspecific incompatibilities. Extensive selfing leads to high homozygous individuals that carry every low survival value. Interspecific incompatibility brings about reproduction isolation, which is responsible for emergence of new races. Self-incompatibility stops free flow of genes, enabling fairly large differences in population. Self-incompatibility prevents self-pollination effectively. As a result, it has a profound effect on plant breeding approaches and objective as given below. The next one is nothing but a self-incompatibility may be used in hybrid seed production. For that, two self-incompatible but cross-compatibility lines are to be interpolated. Seeds obtained from both the lines would be hybrid seed. The next one, self-incompatibility provides a way for hybrid seed production without emasculation and without a restoring of genetic or cytoplasmic male sterility. These are self-sterility as well as a genetic and cytoplasmic male sterility. This is a point we will study in a plant breeding technique. Self-incompatibility system permits combining of desirable genes in a single genotype from two or more different sources through natural cross-pollination, which is not possible in self-compatible species. In case of a pineapple, commercial clones are self-incompatible. Compatible. As a result, their fruits develop parthenocarpically and are seedless one. In case of a self-incompatibility fruit trees, it is necessary to grow two cross-compatible one. How we eliminate these self-incompatibility? For this purpose, we have to first thing remember doubling of a chromosome of that species is important one. Means for example, in them S1 this a single allele is present. The doubling of a chromosome in that case results into formation of homozygous, that is S1 S1. If S1 S2 these two locus these are present, then after their doubling it results into formation of S1 S1 S2 S2, and that doubling is necessary for the elimination of self incompatibility. Identification of self compatible mutant plants from a population of plants, these are developed by some seeds, same seeds. Incorporation of self-compatibility genes of relative species of varieties of some species by backcross method. And these the self-incompatibility, their utilization is done in a plant breeding, and their utilization is done in the production of a hybrid seeds. It is useful in a cabbage as well as a Brussels sprout hybrid seed production. Then hybrid seed production without emasculation and without a restoring of the CMS and GMS, and it is practiced in Japan. For the overcome of that self incompatibility, bud pollination mechanism is important one, and in that case pollination of immature buds with mature pollen grain is done. Then delay pollination, it is a second way for the overcoming of these self incompatibility. In this delay pollination, pollination of aged pistils several days after maturity with normal incompatible pollen is done in that case. Third one is nothing but a late season pollination. In that self pollination at end of a flowering season also lead to the setting of a seed. Then eradication of style with X-ray immediately before selfing result in a breakdown of self incompatibility. High temperature means treatment of a high temperature ranging from 30 to 60 degree centigrade leads to breakdown of self incompatibility. Then multilation of styles means SI could be broken by multilating the stigma with the steel wire brush during the pollination. In vitro fertilization means in that placing the pollen grains in a direct contact with ovules results in a breakdown of cell incompatibility. Thank you, students.